Students, today we are going to discuss about glycolysis. If you see glycolysis, the questions here they can ask the pathway, the steps in a sequential order. They can ask you about the enzymes, or they will ask the energetics in it, and they can even ask inhibitors. Okay, so let's begin our pathway. Now, the name itself, you can tell something. Glycolysis. Lysis means breakdown. Now what is getting breakdown? Glyco. Glucose is getting broken down. Now if you see the basic definition, it is the pathway of conversion of glucose to what? Pyruvate in which thing? Aerobic respiration, and lactate in anaerobic thing. Got it? We will see in the subsequent steps how these are formed. Okay. Now, it is one of the important MCQ that it is the only pathway which is happening, which is operating in anaerobic conditions. Okay, students. Now, let us see the site and state during which glycolysis is happening. If we see the site, it is happening in the cytoplasm of all cells and tissues, including your RBCs. Okay. So what is the metabolic fuel of RBC? It is only glucose. Okay. Metabolic fuel of RBC is glucose. Now, why did I mention the state? During which state it happened? It happens during fed state. So what is the hormone during fed state, students? As you know, yes, it is your insulin. You just remember this word insulin, students. Later we will see the regulation of it, so that whether it is phosphorylating the enzymes or dephosphorylating the enzymes, we will see it in the regulatory step. Okay. Now if you see the whole of the pathway, it is of total 10 steps which is happening in two stages. Okay, the first stage is the energy investment stage. The second thing is energy payoff stage. Okay. So what is happening in this energy investment phase? So bit of energy is being utilized in the starting reactions of the glycolysis, and bit of energy is released in the later part of the reactions. Okay. So here energy is investing in the form of your ATP getting converted into ADP. Okay. Let's go into the first step of glycolysis. In the first step, glucose is getting converted into glucose 6 phosphate. So, so what is the requirement of this step? Let's take an example. Suppose a neighbor of you came to your house. You want to make the neighbor to stay for a long. What will mother do? She will immediately go and bring some snacks and keep it there. So what exactly is happening is we are trapping the neighbors. Okay, in the same way, once glucose enter into the cell through glucose transporter, glucose is getting trapped in the form of glucose 6 phosphate. Okay. Now what are the enzymes which are helping in this thing? It is either your hexokinase or glucokinase. Okay, got it, students? It is either your hexokinase or glucokinase. Here ATP is getting converted into ADP. What is required? Mg2 plus is required. Okay, yes. Now, this glucose 6 phosphate, which is formed, it can go into different things. Either it can go into glycolysis, or it can go into glycogenesis, or lactose synthesis, HMP, or uronic pathway. It has different fates. It can go any of these things. Okay, now we are going to see the difference between hexokinase and glucokinase. So basically, if we see the properties of hexokinase and glucokinase, they are differing in these things. One is the location. Hexokinase is present in all cells, whereas glucokinase is present only in liver and beta cells of pancreas. Okay, now the affinity for hexokinase is high, the affinity for glucokinase is low. Okay, what do you mean by affinity? Okay, if some substance have high affinity means even in the low concentration of that substrate, they are going to, these enzymes are going to act. Okay, let's take the example. Suppose there is glucose of 80 milligram per deciliter. Now hexokinase will act on it and will convert it into glucose 6 phosphate, whereas glucokinase cannot act on this. It requires sufficient higher concentration. Okay, now what about Km? Affinity and Km are inversely proportional to each other. Now, if I am telling you that affinity and Km are inversely proportional to each other, more affinity means yes, less Km. Here it is less affinity means more Km. Okay, very good. Now, what about velocity, students? If you see the basic definition of Km, Km is the concentration at which velocity is half of Vmax. So, Km and velocity are directly proportional. That means hexokinase has less velocity compared to glucokinase, and glucokinase has more velocity. What do you mean by this? If you see in the portal circulation, after taking the food, whatever the glucose which is present there, liver will immediately take up and it will convert it into glucose 6 phosphate very fastly because of presence of glucokinase. Okay, got it? Now we will see the specificity. Hexokinase is not that much specific. It acts on glucose, fructose, and mannose. But glucokinase is specific. It acts only on glucokinase. Got it? Okay. Now, induction. Here it is non-inducible. Here it is inducible by insulin. Got it? Insulin is inducing glucokinase, okay, but not hexokinase. Now, if we see the feedback inhibition, the product of hexokinase itself is inhibiting it. What is the product of hexokinase? Glucose 6 phosphate. There is no such inhibition with glucokinase. Okay. Now, if you see the functions of it, it acts on, mainly the hexokinase acts on the blood glucose level, and it usually is saturated. usually acts on blood glucose level, and mostly it is saturated. Okay, but here glucokinase acts as sensor. Which sensor? Glucose sensor. Got it? So how will it act as glucose sensor? Suppose if you take beta cells of pancreas, whenever the person is in the fed state, glucose will enter into beta cells of pancreas through GLUT2. Once it enter, glucokinase will convert glucose into glucose 6 phosphate, and finally it will enter into the Krebs cycle and it will produce ATP. With the help of ATP, it is going to stop the potassium channels so that the membrane gets depolarized and calcium enters into the cell. Because of that, the insulin will be released. Okay, in this way, glucokinase is acting as glucose sensor. Got it? Okay. This is the just we will revise the first step of um, glycolysis, where glucose is getting converted into glucose 6 phosphate with the help of either hexokinase or glucokinase. And this glucose 6 phosphate has different fates, whether it can go to glycolysis, glycogenesis, lactose synthesis, HMP, or uronic pathway. And we are seeing the difference between hexokinase and glucokinase. There is a difference in the location, affinity, Km, velocity, specificity, and induction, feedback inhibition, and the functions. Okay, now in the next step we are going to see glucose getting converted into glucose 6 phosphate getting converted to fructose 6 phosphate. Okay, what is the enzyme, student, here? Yes, it is phospho hexo isomerase. Okay, so that glucose is getting converted into fructose 6 phosphate. Okay, there is no that much significance about this step, and the next step is very important, where there is formation of fructose 1,6 bisphosphate. What is this enzyme? It is very important enzyme. This is phosphofructokinase 1. Okay, now this ATP and citrate are inhibiting this enzyme. What do you mean by more ATP and citrate in the cell? It means the cell is in high energy states. Okay, so during high energy states, usually after fed state, glycolysis won't be happening, but rather it will go to glycogen synthesis. Now, what are activators of this thing? If ATP is inhibitor, what can be the activator? Yeah, it is your AMP. And what else? It is fructose 2,6 bisphosphate. I will tell you where did this fructose 2,6 bisphosphate coming from. Okay, now here fructose 6 phosphate is getting converted into fructose 1,6 bisphosphate. Okay, and this is going to activate pyruvate kinase enzyme. Okay, activate means allosterically activated. Okay, Now, if you see this enzyme, it is the regulatory enzyme of whole glycolysis, and this step is committed step of glycolysis. What do you mean by committed step? Suppose if someone is telling that they are committed to someone, they will be moving lifelong like that. In the same way, here in glycolysis, once fructose 1,6 bisphosphate is formed, then it will be completing the process of glycolysis. Okay. Before that, as you see, as you had seen, glucose 6 phosphate is taking different routes. But once fructose 1,6 bisphosphate has formed, then it will complete the glycolysis. Okay. Now, this is a feed forward reaction where what do you mean by this? The products are increasing the activity of PFK1. See, what do you mean by feedback inhibition, students? Suppose the products which are formed is going to inhibit the enzyme that is called as feedback inhibition. Now, feed forward reaction means here the products which are formed, it is going to activate the enzyme too much so that it is feed forward, it is going forward. Okay. Now you have two enzyme systems, one is your PFK1 and another thing is PFK2. In PFK2 you have two things, one is the kinase activity and second thing is your phosphatase activity. If you see this reaction, fructose 6 phosphate with the help of kinase activity is getting converted into fructose 2,6 bisphosphate. In this way, fructose 2,6 bisphosphate is formed, which is going to activate PFK1. Got it? Understood? I am repeating. See, you have total of two enzymes. One is your PFK1 and another thing is your PFK2. In PFK2, you have two activities. One is kinase activity and second thing is phosphatase activity. The kinase activity is going to convert fructose 6 phosphate into fructose 2,6 bisphosphate, which is allosterically activating phosphofructokinase 1. And you will see the reverse thing in gluconeogenesis, okay, where fructose 2,6 bisphosphatase or PFK2 phosphatase is acting. Okay. Now, if you see PFK2, it is being regulated both allosterically and covalent modification. We will see the covalent modification in the end of this class, students, when we are discussing the regulation of insulin. Okay. Now, in the next step, there is formation of trioses. So, what do you mean by triose? Very good. It is 3 carbon compounds. Okay. Now, fructose 1,6 bisphosphate is getting broken down into glyceraldehyde and dihydroxy acetone phosphate. What is this enzyme here? Very good. Aldolase A. Okay, don't get confused with aldolase A and aldolase B. Okay. Now, aldolase B is usually present in liver and kidneys and it helps in the fructose metabolism. Okay. Aldolase B is usually present in, yeah, liver and kidneys. Now, from the subsequent reactions, this glyceraldehyde, sorry, it is glyceraldehyde 3 phosphate. Okay. Glyceraldehyde, in the subsequent reactions, this glyceraldehyde 3 phosphate is going to be acted upon. So, this dihydroxyacetone phosphate will also get converted into glyceraldehyde 3 phosphate. Okay, with the help of course, we will see in the next steps phospho triose isomerase. Okay, got it? Yeah. Just don't be confused about aldolase A and aldolase B. Okay. Just be careful when you are attempting these MCQs. Now, in the next step, there is interconversion of these two trioses, glyceraldehyde 3 phosphate is 3 phosphate with the help of phosphotriose. This is isomerase. Okay. This DHAP is getting converted into glyceraldehyde 3 phosphate. From the next reaction, G G3P will be forming the pyruvate. So from one glucose, how many pyruvates are getting formed? Very good. It is 2 pyruvate. Okay. From the subsequent reactions, from the next reaction, glyceraldehyde 3 phosphate getting converted into pyruvate or lactate if it is anaerobically. So therefore, one glucose will be getting converted into 2 pyruvate or 2 lactate. Okay, understood till now? Got it. In the next step, there is formation of 1,3-bisphosphoglycerate or 1,3-bisphosphoglyceric acid. Okay, it is the oxidative phosphorylation step, and it is being done by this enzyme glyceraldehyde 3 phosphate dehydrogenase. Okay, here glyceraldehyde 3 phosphate is getting converted into 1,3 bis phosphoglycerate. Okay, you are telling this as an oxidative phosphorylation. So who is giving this phosphate group? Inorganic phosphate. Okay, is the source of phosphate group here. It is not ATP. Just be careful about this. Okay, and you are telling oxidation. So here NAD plus is getting converted into NADH plus H plus. Got it? Understood? Okay. Now here is the first inhibition thing. Glyceraldehyde 3 phosphate dehydrogenase is inhibited by your arsenate. Okay, just remember this thing. Okay, now in the next step, there is formation of 3 phosphoglycerate. Got it? Now it is being done by the enzyme phosphoglycerate kinase. Okay, where ADP is getting converted into ATP, the help of Mg2 plus. Got it? Here in this step, 1,3-bisphosphoglycerate, it is leaving its one phosphate group to ADP so that ATP is formed, and it is itself is converting to 3-phosphoglycerate. Now there is another alternative cycle here. It is called Rappaport-Leuenberg cycle. Okay, just write down Rappaport-Leuenberg cycle, where this 1,3 BPG is first converted into 2,3 BPG, 2,3 bisphosphoglycerate with the help of the enzyme phosphoglyceromutase. Here it is mutase, phosphoglyceromutase, and this 2,3-BPG will be converting back to 3 phosphoglycerate with the help of this enzyme phosphoglycerate phosphatase. Okay, one phosphate is lost in the form of inorganic phosphate. Okay, what is this significance? See here 2,3-BPG which is formed here, it is required for the removal or giving of oxygen at the level of tissues. Okay, now it is the only kinase reaction in whole of the glycolysis which is reversible. Okay, now ATP is produced at the level of substrate. See, in order for the ATP to produce, the things has to reducing equivalents has to go to ETC and finally ATP is produced. Apart from that, if ATP is produced at the level of substrate, it is called as substrate level phosphorylation. Okay, this is the first substrate level phosphorylation. Okay, now so for one molecule of 1,3-BPG, we are getting one ATP. That means one glucose can produce how many ATPs at this step? Two ATPs at this step. Okay. In the next step, there is a isomerization happening where this 3 phosphoglycerate is getting converted into 2 phosphoglycerate with the help of this enzyme phosphoglyceromutase. Okay. See, I forgot to tell this. See, if in this reaction, if this is going to Rappaport-Leuenberg cycle, there is no formation of ATP. Okay, just remember that point, students. Now, in the next step, there is formation of phosphoenol pyruvate from 2 phosphoglycerate. The enzyme is enolase. Okay, with the removal of one water molecule, so that phosphoenol pyruvate is formed. Now, if someone had asked you in which color vial you are going to estimate glucose, what did you need to tell? In gray color vial. It is gray vial. Okay, let me draw it with gray. Okay, why? What does gray vial consist of? Yes, it consists of fluoride, which is going to inhibit this enolase. See, suppose let's take students, you had collected blood so that you are sending it for the glucose estimation. What is the metabolic fuel of RBC? Glucose. So if you estimate glucose without stopping the glycolysis, it will be underestimated. Okay, so in order to stop this glycolysis process, particularly glucose is collected in the gray vial which has fluoride in it, so that it will inhibit the glycolysis at this step of enolase. This step which is being done by the enolase. Okay, now the last step, it is the formation of pyruvate in aerobic respiration. Okay, this phosphoenol pyruvate gets converted into pyruvate with the help of, yes, pyruvate kinase. Okay, where ADP is converting into ATP with the help of Mg2 plus. Got it? It happens during normal aerobic respiration. Okay, If at all there is anaerobic conditions, this pyruvate will be converting into lactate with the help of lactate dehydrogenase. Okay, where NADH plus H plus, which is formed in the step of oxidative phosphorylation is used up. Okay, is getting converted into NAD plus. Got it? Understood? Now, lactate formation ensures the continuation of glycolysis under anaerobic conditions. What does it means? Suppose there are anaerobic conditions. Now, lactate is not formed. What happens to NADH plus H plus? It is not going to convert into NAD plus. Now, if it is not going to convert it into NAD plus, this NADH plus H plus is going to accumulate. So at this step of oxidative phosphorylation, you require NAD plus. So the reaction won't go forward. Understood? That is the meaning of this line. Lactate formation ensures the continuation of glycolysis under anaerobic conditions. Now let us see the energy production of glycolysis. Okay. Now, in the starting step, I told there are total of two stages. One is your energy investment stage, and another thing is your energy payoff phase. Till the formation of this trioses, the stages whichever are there, they are energy investment phase, and after that, whatever the stages are there, they are of energy payoff phase. Okay. Now we will see how many ATPs are being utilized and produced at different steps. Okay, there is utilization of one ATP at the level of hexokinase or glucokinase thing. Okay, and one ATP is utilized at, recall students, yes, phosphofructokinase 1. And if at all the two NADH plus H plus which are formed, if it enters to ETC via mitochondria, each NADH will produce 2.5 ATP, so that in total how many will be formed? There are 5 ATPs will be formed. Okay. Now there are two substrate level phosphorylations which are happening at the step of phosphoglycerokinase and pyruvate kinase. Got it? So if someone asks what is the net production of ATP in glycolysis, you tell the answer by doing the simple calculation. It is total plus 7 ATP. Okay. So in anaerobic glycolysis, what is the net production of ATP? It is, yes, 2 ATP. The reason is as NADH are utilized in lactate production. Okay. Now we will see the regulation of glycolysis. If you see the regulation, it is mainly based on the ATP demand and glucose availability. Okay, so that is one of the reason. If there is energy rich states, ATP and citrate are going to inhibit which enzyme? The regulatory enzyme PFK1. Okay, now what are the three irreversible steps which are happening in glycolysis? Your the step which is happening in the conversion of glucose to glucose 6 phosphate, your hexokinase or glucokinase step, and phosphofructokinase 1, and finally pyruvate kinase. Okay, now let us see how insulin and glucagon will act on the enzymes and how they regulate If you remember this simple thing, you can write n number of questions whatever they give on your phosphorylation or dephosphorylation. Okay, in this thing first you see whether it is fasting state or fed state. Okay, if it is fasting state, what is the hormone of fasting state? Yes, it is your glucagon. Okay, what is the hormone of fed state? Yes, very good, it is your insulin. Now, how will glucagon acts? So, glucagon will act on G protein couple receptors. Okay, whenever there is increased glucagon in the body, it will act on G protein coupled receptors, the GPCRs. Okay, now it is going to activate alpha subunit of GPCR. See, GPCR has three subunits in it: alpha, beta, and gamma. Okay. Now, once suppose it is a cell, and here is your GPCR receptor, which is like a serpentile, and it crosses 7 times. Now, once glucagon comes and act on it, here to the inner side of it, you have alpha subunit, beta, all this thing. Okay, and once glucagon comes, it is going to activate alpha subunit of GPCR, and then this alpha subunit is going to activate adenyl cyclase. Now this adenyl cyclase converts ATP into cyclic AMP. Okay, it is going to activate protein kinase A. Very good. Now this protein kinase A phosphorylates all the enzymes. Remember this word, phosphorylation. Glucagon phosphorylation, insulin dephosphorylation. So glucagon will activate the enzymes of fasting state by phosphorylating it. Okay, it will phosphorylate and it will activate. Insulin will activate the enzymes of the fed state by dephosphorylating it. If you remember this point, you will do n number of MCQs. Okay, now whatever the enzymes which need phosphate for its activation, they will get activated. Let's take the example. The classic example is glycogen phosphorylase is activated by phosphorylation with the help of glucagon. Okay, now if it is of fed state, what is the hormone of fed state? Yes, it is your insulin. Now, how will insulin works? So insulin will activate phosphodiesterase, which is going to degrade your cyclic AMP. Got it? Now it will activate protein phosphatase, which is going to dephosphorylate all enzymes in fed state. Got it? Okay. Now let us see few MCQs based on this class. Okay. Which of the following is the rate limiting enzyme of glycolysis? You know it, students, very well. It is your phosphofructokinase. Okay, it is option B. Now, in the Rappaport-Lewinberg cycle in RBC, which intermediate is bypassed? Okay, they are asking what is the intermediate which is being bypassed in uh, Rappaport-Lewinberg cycle. You find it out. 1,3-bisphosphate glycerate. No, it is the starting substance. 3-phosphoglycerate. It is the ending product, ending step. Phosphoglycerate kinase step. Yes, obviously. Okay. This is what it is missing. Okay. Now, fluoride inhibits which enzyme of glycolysis? As I told you. Which enzyme, students? Yes, it is enolase. Very good. Now, how many net ATP molecules are produced during anaerobic glycolysis? They are asking, see the question clearly. They are asking anaerobic and the net. How many it is? It is 2. Okay. Now, which of the following is a specific inhibitor of glucokinase? What is the specific inhibitor of glucokinase, students? Glucose regulatory protein. I didn't discuss this thing, but when we are dealing with the genetics, we will discuss it. Okay. Now, these are the new model MCQs. You just try it. Arsenate poisoning affects glycolysis by competing with which substrate? See, usually arsenate will inhibit that enzyme by competing with the inorganic phosphate which is present in it. Okay, got it? Yeah. Now, because arsenate will mimic as inorganic phosphate and it will go and it will inhibit the enzyme. Got it? Okay. Now, maturity onset diabetes of young type 2 is caused by a mutation of which enzyme? Okay. Now, what is the enzyme which is acting as glucose sensor? Yes, it is your glucokinase. So, glucokinase usually will act at more 130 milligram per deciliter. But in there is any mutation in glucokinase thing, it will act in more amount which is like greater than 140 milligram per deciliter, so it will lead to this disease like maturity onset diabetic of young type 2. Usually it is not noticeable, but if sometimes particularly pregnant ladies, if they go, they will definitely check for the sugars and unknowingly it will be found out. Okay, it is your glucokinase. What is the which of the following is the most potent allosteric allosteric activator of PFK1? You can tell it, students. What is the most potent allosteric activator of PFK1? Yes, it is your fructose 2,6 bisphosphate. Now see it. Hemolytic anemia in pyruvate kinase deficiency is primarily due to. See, I didn't discuss this thing, but you can guess it. Okay. See, our body requires sodium potassium pump continuously. For sodium potassium pump to work continuously, it requires ATP continuously. If there is any issue with the pyruvate kinase, it can't complete sorry, it can't complete the ATP production, all these things. Okay. So that there is hemolytic anemia. Got it? Okay. Why particularly pyruvate kinase? If you see pyruvate kinase step, there is a formation of substrate level phosphorylation. Okay. Now, in the liver, the bifunctional enzyme PFK2 and fructose bisphosphatase 2 is dephosphorylated in response to what? So here, see it properly. They had used the word dephosphorylated. So insulin. Got it? Understood? Any doubts? Okay. Thank you.